Hi, in this lecture we're going to be introduced to three specific types of signaling pathways: ligand-gated ion channels, G-protein coupled receptors, and receptor tyrosine kinases. These are the three major classes and types of pathways, and it's important to be introduced to each. You may feel at times overwhelmed by a lot of the details specific to each of these pathways, but it's important at least to get exposed to these terms to facilitate your future discussions. In the course of discussing these pathways, we'll encounter other ways in which information is communicated inside the cell. You'll recall that the primary method is the phosphorylation of proteins, but cells can also use small molecules like cyclic AMP or phosphorylated sugars to transmit information, and we'll see examples of those. This is a very brief review slide reminding you that signaling occurs when an extracellular signal molecule binds to receptor proteins sitting on the surface of the target cells. These receptors act as signal transducers by undergoing a conformational change and also inactivity, triggering a set of downstream events that transmits the signal intracellularly to alter the behavior of the cell. Ion channel coupled receptors are the simplest of all the signaling pathways, and we can cover it pretty quickly. In this case, a signaling molecule will bind to a receptor, causing a conformation change. In this case, it usually causes ion channels to open, allowing ions to pass through. These types of pathways are heavily involved in the nervous system. For instance, at the neuromuscular junction, where nerves couple to muscles to produce a process called excitation-contraction coupling. An action potential in the nerve releases the ligand, which in this case is acetylcholine, which diffuses through the synapse and binds to the receptor molecule on the muscle cells, causing the acetylcholine receptor to open, sodium ions to flow in, and muscle contraction to result. The contraction stops when a protein called acetylcholine esterase degrades the acetylcholine in the synapse, causing the ion channels to close. There are also ion channel coupled receptors for other ions, including potassium, calcium, and chloride, and other ligands include serotonin, a neurotransmitter, and GABA, also a neurotransmitter, short for gamma-aminobutyric acid. The second class of signaling pathways are referred to as G-protein coupled receptors. First, a point about nomenclature. Here is the receptor, and here are trimeric G proteins. Trimeric referring to the fact that there are three subunits. These G proteins are functionally coupled to this receptor, giving the name G-protein coupled receptors. You can remember this abbreviation as it also contains the letters PCR in that order, the important nucleic acid amplification technique. And we'll see this in more detail in the next slide, but a binding effect of a signaling molecule to the receptor produces a conformation and activity change in the cytoplasmic tail of the receptor. This causes the trimeric G proteins to become associated with the receptor, and it activates it, removal of GTP and the incorporation of GTP. Upon being activated by GTP, the trimeric proteins dissociate into two subunits, the alpha and the beta gamma, and they diffuse laterally tethered to the membrane by the lipid tails, which are covalently attached to the proteins, to an effector protein which then they can activate. This is an example of the activated beta gamma complex interacting with a potassium channel, causing it to open and allowing potassium ions to flow out. Eventually, the GTP in the alpha subunit will hydrolyze to GDP. This will increase the affinity for the complexes again, causing them to reassociate and inactivating the beta gamma complex as well. Another class of G-protein coupled receptors are those that use as intermediates the molecule cyclic AMP and the protein kinase A. In this case, the binding of the signal molecule activates the receptor, activates the trimeric G protein, of which thus the alpha subunit is shown. This will activate adenylyl cyclase, which is an enzyme which will take the molecule ATP and produce cyclic AMP from it. We've seen cyclic AMP before, and I'll show you the structure on the next slide. But for a period of time, this cyclic AMP molecule is the carrier of the information that a binding event has occurred on the outside. This cyclic AMP molecule will eventually be bound by protein kinase A, causing a conformational change, causing it to be translocated into the nucleus, where it will phosphorylate a transcription factor. The phosphorylation of the transcription factor allows it to change a conformation, now making it suitable for binding DNA. It will turn on transcription of a gene with the product of an RNA molecule, later to become a protein molecule and constitute a response to the extracellular signal. So the information flow is the binding of a signaling molecule, change in conformation and activity of the receptor, the binding of GTP to the trimeric G proteins, activation of an enzyme, a rise in the concentration of cyclic AMP, activation of a kinase protein, phosphorylation of a transcription factor, the production of an RNA and protein. This slide is just a reminder of the structures. Here's ATP with 3 phosphate groups. The enzyme adenylyl cyclase will cause one phosphate group to bridge the 3' and the 5' carbons, causing this cyclic structure, which is the active signaling intermediate. Eventually, signaling stops when cyclic AMP is cleaved by the cyclic AMP phosphodiesterase. On this slide, we see another complex G protein coupled receptor pathway, which uses a variety of mechanisms for transmitting information inside the cell. The story starts again in the usual way: a signal molecule binding to a G protein coupled receptor, a change in conformation and activity of the cytoplasmic tail, the activation of trimeric G proteins by the binding of GTP, and then these proteins go on to produce unique downstream events. In this case, the alpha subunit is going to activate a protein called phospholipase C, which is an enzyme which has an unusual activity. True to name, it's phospholipase. Here's a phospholipid called inositol phospholipid, which consists of the sugar inositol and a lipid, a diacylglycerol. And it's going to cleave this phospholipid into 2 parts. Here you see the diacylglycerol, which is obligated to stay inside the membrane, and this now free to move in the cytosol sugar, inositol triphosphate. We'll see on the slide that inositol is a 6-carbon sugar, just like glucose, with unique stereochemistry of the hydroxyl groups. In this case, 3 of the 6 hydroxyl groups are phosphorylated. So one of the very interesting things about this signaling event is that both the parts, the diacylglycerol and the inositol triphosphate, are active signaling intermediates. Inositol triphosphate will bind to this calcium channel present on the surface of the endoplasmic reticulum. The lumen of the ER has high concentrations of calcium, which are now, when the channel opens, allowed to go out into the cytosol. Normally the concentration out here of calcium is very low. Now it's increased. The increased calcium can bind to this protein called protein kinase C. But calcium binding it alone is not enough to activate it. It also binds to the diacylglycerol, which was liberated by the cleavage of this phospholipid. So both parts, the diacylglycerol and the calcium ion, activates protein kinase C. Now this goes on to phosphorylate other proteins in a downstream manner in order to affect a cellular response. This slide shows some of the molecular details of the inositol phospholipid, which gets cleaved here, resulting in the production of the diacylglycerol part and inositol triphosphate. You remember about triglycerides. We have the backbone glycerol molecule, of which 2 of these are attached to fatty acids, part of the membrane, and that 3rd hydroxyl group was connected to this phosphate group of this sugar. Here we can see the stereochemistry of the 6 carbons. These hydroxyl groups are phosphorylated by kinases which act on sugars, just like kinases can also act on proteins. And there are also phosphatases specific for the removal of phosphate groups from these sugars. These structures are regulated because they have specific functions. And again, this one helps open the calcium channel in the ER. Okay, and here will be a good place to pause before we start the third type of signaling pathway. Thanks for listening.